Dr. Corida Grudzen joins us now, and she's with NYU Langone. Dr. Grudzen, thank you so much for your time.
Thank you for inviting us.
Well, what do you hope to prove with this study?
So what we're doing is a randomized controlled trial, as you said, of convalescent plasma, and it's comparing it to a control group of normal saline. So that's basically doesn't do anything. So what we hope to see is that convalescent plasma used at this stage of disease, so early on, prevents patients from dying, from going on a mechanical ventilator, or any sort of bad outcome. That's really the goal.
Okay, now convalescent plasma, that kind of treatment has been around for quite some time, actually since the early days of fighting polio. Can you explain briefly how it works?
Sure. So the first Nobel Prize in medicine was actually given for this process to an immunologist who developed a process for taking convalescent plasma and giving it to children with diphtheria. So that was the first Nobel Prize in medicine and physiology, and it's called passive immunity. So vaccines are active immunity. You get an injection, your body has to make the antibodies and prepare itself. Passive immunity is basically borrowing antibodies from someone who was sick and is now convalesced or feeling better, and putting it in their blood so that they don't have to make the antibodies because there's no time. So it's sort of a stopgap measure in the sense that when you don't have a vaccine, it's something that can be used in a new infection where we don't have known drugs or other therapeutics or biologics that we know that can work against the disease.
Now, who is taking part in the study? If it's not a blinded or controlled study, how will you identify the benefits since there may be a wide variation among treated subjects?
Yeah, so it is blinded and it is controlled. And our inclusion, so we're looking at adults, patients 18 and over who are hospitalized. And we're getting them early in the course of their illness. So the first 72 hours of hospitalization, or if they've been ill for less than 7 days. And the reason we chose that population, there are many other studies across the country that look in different populations, but I'm in New York City working in an emergency physician. Many of us were seeing hundreds and now thousands of patients hospitalized. And so we really wanted to focus on that group. We were just seeing in front of us these terrible outcomes in hospitalized patients, and we wanted to see if this could prevent that sort of event. So that's why we chose this population, and we know historically what it does is it neutralizes. So the antibody binds to the virus in some way to prevent it from either entering the cells or for destroying whomever it's attacking. And so the idea is you want to do that before there's an onslaught of the body's own inflammatory response.
Okay, so it sounds like timing is key here. I mean, do you have an idea?
That.
Yes.
Yeah, we
okay.
do believe that, but we'll find out because there are other randomized controlled trials across the country. There's one at Hopkins that is so early, it's giving it to people at risk for developing the disease, but who aren't even ill yet.
Okay.
We felt strongly we wanted to test it in hospitalized patients. There are some who are not on a breathing machine or mechanically ventilated. There are other sites that are testing it in that sicker population. But we, in talking to experts around the country, we know that the earlier the better. And when you're already in the ICU, it's not so much the virus that's destroying the body at that point, it's the inflammatory response to the virus. So it's things like your body's own overreactive immune system that's causing problems with the lungs, the kidneys, all of that. So the hope is that, what we believe theoretically, and, we never know until we get the results, is that in the ICU type patient, other drugs may work, but probably convalescent plasma we don't believe will be helpful. But we'll find out from these other studies.
Now, do you have an idea of the sufficient amount of antibodies that you would need from the plasma in order for it to be effective? For instance, is there an exponential decline in antibodies over time after a patient recovers?
Those are great questions. I'll take the first one, which is about, do we know how much? We really don't. What we do know is, number one, that most people that are now convalesced, so that are, proven by a PCR test generally to have had coronavirus and are now feeling well, that almost all of them, if given enough time, so weeks later, and by 28 days, almost everyone has made antibodies. So that's reassuring. How much we need to give? Plasma is given in units. So we give 1 unit in the trial. We may end up needing to change that or test different variations in the future because it may not be enough. We may need 2 units. So expanded access, which I'm sure many people are already aware of, is that protocol is out of Mayo, and that is 1 or 2 units. The issue is, at the beginning of the epidemic, there was no convalescent plasma because no one was convalesced. Everyone was either still ill or they couldn't donate. They were so ill they couldn't donate. So at the time we started, we had very little plasma. So it was not realistic to think we could use 2 units.
Right.
Now that it's, the flow is, there's been a lot of people that have donated. There's plenty of plasma for all the patients that need it. So now we're thinking through, well, do we want to add an additional future evaluation of whether it needs to be 2 units? Right. But our co-investigator at Einstein has tested some of the plasma that's been donated and has found that there's overwhelming amount of antibody in that convalescent plasma. So that's reassuring. Okay.
Now, there seems to be a wide variety of treatments going on across the country. How do you tease out some of the other treatments that have been used, like azithromycin or Plaquenil? How do you know that it's definitely what you're working with and not some of these other medications that have been given to patients?
So, great question. The reason that I am so in favor of randomized controlled trials is that flip of a coin controls for all of that, because basically when you do a randomized controlled trial, that's what we call, the highest level evidence in medicine. And the reason that's so important is because when you randomize, that single act of randomizing patients before you know what they're going to get controls for all of that, because anyone who's randomized, whether they're on those other agents, it'll be split between the 2 groups. So if half of your population is on hydroxychloroquine, that's okay, because it'll balance each other out by that flip of a coin. So we do have patients that are on other agents, but because of that flip of a coin, it's really a computer that does the flip. But because of randomization, it allows us to evaluate things in a much more robust way than just what we call observational research, which can also answer a lot of questions.
Right. Yeah. Well, now, how could what you learned from this study be helpful to those working on a vaccine?
So I think it's not so much helpful to working on a vaccine so much as a stopgap. Ideally, well, I guess there's 2 things. So a vaccine is preventing you from getting ill, whereas convalescent plasma is treating those who are ill. So I think they are absolutely, even if we have a vaccine, if convalescent plasma is found to be effective, there is still a role potentially if people are getting ill or weren't vaccinated, etc. But it's generally a stopgap when you have a new disease, so a disease that's not been seen in the population before, such as, at the time, diphtheria, the Spanish flu, other diseases, we've seen SARS. So that's the idea, is that we don't have any known therapies that are effective.
Now, what's the downside? Any risks to this type of infusion?
Absolutely. So sometimes patients and families aren't always aware of the risk. So, we read about it a lot in the news. And so there's a lot of discussions that happen at the bedside, because I think what people don't know about is the potential harms of plasma. So plasma is a blood product. So like any blood product, the two main potential harms are, number one, getting an infectious disease. So of course, the blood is screened for all of those things, but like we know about, coronavirus 19, there are new diseases all the time you aren't screening for. And so that's a potential harm, is you can get an infectious disease. The second one, there's many, but the second big one is a reaction to the plasma itself. So once again, it's a blood product. It's coming from someone else into your body, and your body can have from a minor allergic reaction where you just get an itchy rash, we give you Benadryl, to an overwhelming response which shuts down your organs. And especially in older people, it's a little more risky, because those you can have what's called transfusion-associated lung injury. You can have all sorts of damage to kidneys, to lungs, to the heart, etc.
Right.
So it's not without potential harms, which is why we have to have very long, lengthy discussions with patients and families about balancing the potential harms and the potential benefits, and making sure that they understand and have realistic expectations for what the plasma may or may not do.
So in terms of hospitals administering this, how do they decide who can try it and who can't?
So in a randomized controlled trial, it's very easy. The criteria are very objective. So like I said, you have to be 18 or over, a certain phase in the illness, etc. Now, there is no FDA approved approval for convalescent plasma, so it's an investigational new drug. So all of it is under the regulation of the FDA. So the FDA, for the expanded access, which is outside of a trial, they also have criteria. So it's highly regulated. So those criteria are a little bit looser. There are things like severe life-threatening illness. They're not as quantifiable, like 72 hours of being ill. So that gives us a little more leeway as physicians at the bedside to use it, even though patients may not fit the trial. We can still, even if they're excluded from the trial, we can still use it under this expanded access protocol through the FDA and make those kind of decisions, as long as we think the patient is more likely to benefit and less likely to be harmed by giving that plasma under that other protocol.
Oh, okay. Now, plasma therapy is generally known to be helpful in fighting off some viruses, but maybe not so much in others. I mean, why is that?
So I think we have all these theoretical things we can say in medicine. Well, theoretically, it would work in this case or that case, but really the proof is in that gold standard of a randomized controlled trial. So we've seen it work in other viruses but when we say we've seen it work, there actually have been no randomized controlled trials. So there's been studies where they kind of match. They say, okay, this patient got it, this patient did it, these two patients are similar. We see the patients who got it seem to do better. But as you know, as everyone knows, there are reasons we give plasma or other agents to certain patients and not to others. So they might be sicker, so we give it to them. They might have, all sorts of things, something to do with their kidneys, their lungs. And so the patients, unless you use that key, that randomization, that flip of a coin to decide whether to give the plasma or the therapy, it's hard to really know. Another key consideration in whether it works or not is there neutralizing antibody. So there may be plenty of antibody, but it may not actually be the neutralizing antibody, which is the key to preventing poor outcomes. So we don't honestly know that yet. We know that there is plenty of antibody in convalescent plasma. We don't know much about the neutralizing antibodies specifically. So that may be one reason it may or may not be effective. And, as I'm sure you're aware, there's a certain amount of time, and we don't know this for this disease, but when antibody may wane in people who are convalescent. We don't know the answers to any of those questions yet, unfortunately. We're working 24/7 to try to find out, but it's still a lot of unknowns.
So what is the timing of this? How long will it take to complete this study?
So that's another unknown, unfortunately. So the good news is we have less patients in the hospital with coronavirus. The bad news is that means we're having trouble finishing our research because we rely on, infected patients to know if things are working or not. So, of course, that's wonderful for human health that we have less people getting ill and hospitalized. But the problem with research in infectious disease is precisely that you have a pandemic, you don't know anything about therapeutics or biologics or what works, what doesn't work to improve outcomes. And then right when you start the trial, you weren't able to finish it because with the trial, we have to reach 300 patients. We have over 100 now, but we're not even at the halfway point. And so if there's not enough people to enroll, then we don't know if it's effective or not because we do something called a power calculation. So that is basically we have to have a certain amount of people to know truly if there's a difference or not. And so I'm sure you saw some of the studies in China were stopped early and we never got an answer because they didn't accrue all the patients.
Right.
So we would be thrilled to recruit, we were trying to recruit. We opened the study at NYU on April 17th. The first few days, we were working around the clock basically to get as many patients enrolled as possible because we felt strongly that we knew this was going to wane. And then, God forbid, the next outbreak comes, we don't have any answers.
Right.
So we really want an answer. So we're trying our best. But at the same time, I would love if we could stop the trial and be done because we got rid of the disease.
Right.
And I think, obviously, our first priority is getting people well and not having any outbreak again. But
right.
we want to be prepared.
Now, if the study is completed, what do you think or what do you hope the broader impact of the therapy will be?
Well, so
if it works.
Completed, and it shows harm, obviously we would hope, that there would be, we always do, what we recommend is even when you see results in one trial, it needs to be repeated. So even if the study shows benefit, harm, or no impact, there will be future studies. So it won't be definitive, but it will be a lot of information, number one. So it may be it's in the wrong population. We didn't use, enough. Maybe it needs to be 2 units instead of 1 unit we're using. So there will be other questions. If it's harmful, that's really important, however, because we'll definitely want to examine a little more about is it harmful in certain patients that are at high risk for, that lung injury I was talking about from plasma. If it's beneficial, I think that's also incredible information because should we have another outbreak, we now have plenty of plasma, so this would be a really viable way to treat people as a stopgap. We would never want this to be the only treatment because it is a blood product. And the potential harms of any blood product are real. And so ideally, I think we would all love a vaccine, obviously. But this is something we can do in the meantime.
Well, since there is so much plasma available, would you consider, or has it been considered, administering this to frontline workers, people who are particularly vulnerable, maybe people who are sick at home and want to get well at home?
Yes, absolutely. And Johns Hopkins has a protocol for exactly that. So giving it to people not outside the hospital setting before they're ill, so people that are high risk for contracting coronavirus. And that is open and enrolling now. So that'll be really interesting to see what they find there as well.
Okay. Dr. Grudzen, is there anything that I've missed?
No, No, I don't think so. Thank you so much for having me on.
Dr. Corida Grudzen, thank you so much. And she's the Vice Chair for Research at the Ronald O. Perlman Department of Emergency Medicine at NYU Langone Health. And I should also mention that Montefiore Hospital System and Albert Einstein School of Medicine were also involved in this work. Thank you so much, Dr. Grudzen.
And thanks to the team of people that have been working on this across both institutions. Thank you.
And I'm Mabel Jong for Health Day Live. Thanks for joining us. See you again.